So I have only recently started working in genetics with Chiari, but I think we found some interesting things already working with Dave Limerick at WashU. So we sort of got into this serendipitously. We are historically a scoliosis lab, so we've done the genetics of idiopathic scoliosis for a while, and we kept seeing patients with Chiari malformations, and so we teamed up with Dave Limerick, who unfortunately had to go to give another talk, but and have collected a large number of patient DNAs and have done a lot of sequencing, and in collaboration with Doug Brockmeyer and others around the country have collected a large number of patients, and I hope we have some interesting results. So, so quickly I'll go through sort of what we currently know about the genetics of Chiari and Syringomyelia as it relates to Chiari. And some epidemiological work that we and others have been doing. So we have about 500 patients at Children's Hospital in St. Louis and at WashU who we have a lot of clinical data and MRIs on, and we've been measuring a lot of things to try and correlate with clinical outcomes, and then hopefully inform the genetic studies that we do. And then I'll tell you a bit about the studies of exome sequencing in Chiari patients that we've been doing and the associations that we've done. I know I'm not talking to an audience of geneticists, so I'll try and explain things that I think might be specific to geneticists, but I think we found some interesting things with variants that are common in the population, some extremely rare genetic variants that we see in Chiari patients, some work that we've done in large families where Chiari segregates in the family, and then we have about a handful of trios now that we've sequenced, parents and an affected child, where the parents have been tested via MRI for Chiari malformation and the child has it, but the parents do not, and we've been looking for what we call de novo variants, so variants that are new in the child, and hoping to find variants that are associated with Chiari in those patients. And then finally, I'll sort of peek into what we're going to start doing now that we have some associations with Chiari malformation in this cohort with our knowledge of zebrafish genetics and some in vitro studies that we like to do as geneticists as well to understand the genes underlying these disorders. So quickly, just a real brief introduction to sort of what we do and what we think about as geneticists of human disorders is we generally like to get a lot of patients, as many as possible, thousands if possible, and then compare their genetics to a very large cohort of controls. And then by doing so, we can find new variants never before seen, and also associate common polymorphisms in the population with the trait using statistical approaches. And then hopefully what we can do is try and understand those variants and the genes that we find associated with these traits in things like zebrafish. We do zebrafish genetics in our lab, but of course many others use mice and so forth. And hopefully to understand the pathophysiology of the genes that we find associated with these disorders, in this case Chiari and Syringomyelia, to help understand the pathophysiology in humans and possibly help the patients understand whether or not they should have surgery, what kinds of symptoms they might have. So there's been a lot of work to understand whether or not Chiari is genetic. This is a good question that human geneticists often have given to them, is this thing even genetic? Well, as a human geneticist, I would argue every single thing in the world is genetic. But the question is, how genetic is it? But there has been a lot of work showing that the rate of Chiari and Syringomyelia are higher in MC twins compared to more distant relatives. There's lots of familial clustering. I'll show you an array of pedigrees that all segregate Chiari And of course, as been discussed a lot in this meeting, is the overabundance of Chiari malformation among people with connective tissue disorders, craniostenosis, neurofibromatosis type 1, etc., suggesting that these genetic disorders clearly lead to an increased risk of Chiari, thus making Chiari itself genetic to some extent. But not that much has been done to determine the genetics of these disorders. There have been a number of groups that have looked, but honestly the sample size that's required to do these sorts of genetic association studies has not been had in these cases. And so our hope was to expand this and find some new things with larger cohorts. And now again, as I discussed earlier, our sort of stumbling in on the genetics of Chiari was that we studied scoliosis in our lab, and there actually is a very striking connection between scoliosis and Chiari and connective tissue disorders. So one of the, and specifically with Syringomyelia. So that's sort of how we got into this. But of course there were also, as I mentioned again, a lot of genetic disorders that have Chiari malformation as a sort of symptom. Most of them are listed here that I could find. But I imagine there are even more. And a lot of these are, in fact, as was mentioned, connective tissue disorders. So in our sort of retrospective cohort of people who had MRIs and were diagnosed with Chiari malformation, which I think is one of the largest currently around, of 500 patients, we found this sort of array of secondary comorbidities, or primary comorbidities among people with this disorder. And a lot of them were central nervous system disorders, genetic disorders, multiple congenital anomalies, or skeletal disorders. And then sort of where we went for genetics is take this big yellow section, all the things where we can't really explain why a person has a Chiari malformation, and try and find genetic contributions in that subset. And then as a sort of tangent, we took that same data set and we looked at a number of MRI measurements, and knowing that Chiari 1.5 is largely determined by Obex position, we actually looked to see if Obex would be able to predict various clinical correlates in this large Chiari cohort. And we saw that it was better able to predict whether or not the person would have surgery. Of course, this is a sort of a not objective test of severity. But individuals with more descending Obex were more likely to have surgery. And as you can hopefully see in this scatter plot, when you correlate tonsillar herniation on the y-axis versus Obex on the x-axis, even controls having 0 tonsillar descent but large high Obex, not low Obex, that the majority of the surgical cases, in fact, had both very low Obex and very large tonsillar descent, suggesting that in addition to tonsillar descent, we should actually be also measuring Obex in all of our patients to determine the severity of their Chiari. And some more data for the relationship between Obex and severity. We actually see that individuals with mild, so lower than the mean tonsillar descent, and yet lower than the mean Obex, so here mild tonsillar descent and severe Obex, actually did far worse. So this is actually Syringomyelia probability. So in our cohort, only the individuals here that had a low Obex, even if they had a very high, large tonsillar descent, showed risk of Syringomyelia in this population. And furthermore, Obex actually predicted occipital headaches versus non-occipital headaches, was correlated better with CSI grade and canal angle in this population as well. So together, that sort of epidemiological data made us actually do a subset genetic analysis with what we are calling Chiari 1.5, but technically is only individuals with low Obex and, at least in our case, 10 millimeters tonsillar herniation as, again, kind of an arbitrary cutoff. This was our aim at getting severe Chiari Malformation patients. And so what we did is exome sequencing, so capturing of the exon coding portion of the genome in a large number of CM1 patients, and in a large number of cases, some affected and unaffected relatives to look at segregation of variants that we find, and a very large number of control individuals, though these are just population controls, so some proportion of them likely do have Chiari, though it would be the proportion that is probable in any individual, which is relatively low. And so we did a standard pipeline to call variants from this exome sequencing and called more than 3 million variants across this cohort in the exon coding portion of the genome. And this is the first genetic results I'll show you, is all variants that occur at a frequency of more than 5% in the population, and their associations using just a normal chi-squared test, comparing the probability of that variant occurring in Chiari versus non-Chiari patients. And what we can see is a number of interesting genes come to the top, some of which may be only interesting if you know scoliosis literature, but so we actually have an association with SLC39A8 in scoliosis, and this is another manganese and zinc transporter, SLC39A3, that's associated here with Chiari specifically. And I'll go into more detail, but we also see a number of CHD genes, which are actually developmental delay genes with, in many cases, dysmorphic features that I think maybe explain the Chiari in this situation, which is chromodomain gene 1L. And then FKBP4 is actually very similar to the FKBP14 gene, which is a known cause of EDS. So together, we're finding some interesting preliminary results with common variants in this cohort. Of course, the issue that human geneticists often find themselves in dealing with is the fact that when you sequence an individual, the vast majority of genetic variation is extremely rare, mostly unique to an individual. So being able to figure out what those specific variants that you're finding in these patients are doing, it can be very difficult. So here's just a histogram of variant frequency in the population, and this is the proportion of polymorphic sites that we observe in our cohort, and the vast majority of these are extremely rare. So this diagram is meant to show basically that it's difficult to deal with these variants. So often rare variants, super rare variants, have very little ability to statistically determine their association with a trait by themselves. Even those that have extremely high effect size that cause Mendelian diseases can be difficult to deal with. So one of the techniques that we've used in the past and have now expanded to our analysis of Chiari patients is this sort of gene burden approach. So what we do is we collapse all of the extremely rare protein coding altering variants that we find in our cases and controls in each gene in the genome, so each test is a single gene, and determine the difference in frequency of that collapsed minor allele frequency in our cases compared to controls. And we did this successfully to discover the association between variants in the FBN1 and FBN2 genes, which respectively cause Marfan syndrome and contractural arachnodactyly in our scoliosis patients. So these patients actually do not have Marfan syndrome or contractural arachnodactyly, but do have scoliosis. And more recently, we found an association, again, in scoliosis with rare variants in a subset of collagen genes, those that cause musculoskeletal Mendelian disorders. And we see much higher frequency of rare variants in our AIS cases compared to controls. So our aim was to do this sort of same analysis with our now growing number of Chiari patients and controls. And so this is the results. So I know I just showed you a graph like this, but so this is a QQ plot for anyone who's a statistician. On the x-axis, we have the expected p-value, negative log of the p-value, and on the y-axis, we have the observed. So the more off the line you are in the up direction, the better, because those are significantly associated variants, or in this case, genes. And what we see is a number of interesting genes coming up. PAX4, which is a homeobox gene. CHD3, again, is a chromodomain gene involved in various developmental disorders, some of which have, again, skull abnormalities. And then filamin B, which is also interesting because of the skeletal disorders that are caused by mutations in that gene. And then I subsetted the data by either all patients with Chiari 1.5, here defined as simply having an Obex less than 0, or Chiari and Syringomyelia. And what we see is actually interesting results given these subsets. So in the case of the black dots, which is all Chiari, we see CHD3. And we also see CHD3 actually coming up to the top in Syringomyelia specifically. But when we subset on patients with Chiari 1.5, we actually see a different subset of CHD genes, which all point to this leading hypothesis that these CHD genes may be involved in Chiari and Syringomyelia. So this is just a blow-up of one of those results, the CHD3 gene. What we see is these rare missense variants occurring in our Chiari patients and not in our controls. So we see about 9 patients with rare, these are all actually unique to these individuals, never before seen in human populations to date, compared to only 2 in our control cohort. And so this gene actually does not have a very a Mendelian disease associated, although other CHD genes do. It's very highly expressed in the brain. It's particularly in the cerebellum, interestingly. It actually cross-links with a gene known to cause cleft lip and palate, so who knows what that means, but it could mean that it's involved in facial development. And interestingly, some common variants are associated near this gene, but again, how that relates to Chiari is yet to be determined, if at all. And so I was actually interested, given that we see a number of these CHD genes at the top of our list for various subsets of our Chiari patients, what the results were for all of the CHD genes. So in total, there are 9 CHD genes in the human genome. 3 of them cause nondescript developmental disorders. 1 causes Charge syndrome, and 1 has been associated with autism, though I would argue most of the time these cohorts of autistic individuals also have developmental delay. So in general, these are developmental delay genes. And what we see is that if you sum up all of the rare variants across this cohort of this subset of genes, we actually see twice as many individuals in our Chiari cases with variants in at least one of these genes compared to our control cohort, which is very interesting. And I think what we really need now is larger cohorts and replication and more families that we can look at these variants and see if they segregate with Chiari. So these are just a subset of our larger Chiari pedigrees from WashU. We have a number of large pedigrees in collaboration with Doug Brockmeyer from the University of Utah, and a number from the University Hospital Vall de Hebron in Barcelona, in collaboration with Alphonse Macaya. And so actually several of these CHD genes actually appear to, at least at the outset, segregate with Chiari in these families. So 2 CHD3 mutations segregate, at least in the patients that we have sequenced. And so going forward, what we're going to do is recontact these families and expand the number of people that we have sequenced. genotype some of these variants in the rest of the families. Lastly, one of the techniques that we can use in human genetics to identify interesting genes in disorders, especially those that have early onset with severe, in this case, usually this is best for very severe disorders that have reproductive consequences. But I would argue if you had extremely severe Chiari malformation that led to breathing problems and death in the past, you could in fact have a depletion of mutations in some genes that would lead to this phenomenon in Chiari as well. And so we've sequenced now 17 trios where the parents are confirmed MRI negative for Chiari malformation and the child is MRI confirmed for Chiari malformation. And in general, when we do exome sequencing in these cohorts, we see 1 to 4 rare de novo coding SNPs per family. So it quickly narrows down the set of genes that are important if you have enough of these sorts of trios. And so we've been doing a lot of work to collect parents, if possible, for children with CM1 and to try and do this sort of analysis in as large a cohort as possible. So this is sort of the tip of the iceberg in our 14 trios for looking at de novo variants. Interestingly, we actually see a de novo stop codon predicted to be pathogenic in a CHD gene, so CHD8. And we actually have Sanger sequenced this and found it to be true and real in this patient. And so we think, although this patient does not actually have autism, they do have growth delay and developmental delay in that way. So going forward, using these association results, our next sort of plan of attack is to try and understand whether or not some of these genes do in fact lead to Chiari-like phenotypes in model organisms or affect the protein function in vitro. So the first, so this is just a sort of a snippet of things that we and others have found to be measurable in zebrafish that are related to Chiari. So you can look at hydrocephalus at very young ages. You can actually stain ventricles in the developing zebrafish and measure their size, for instance. Actually, CSF flow has been recently associated with scoliosis in zebrafish, and so it'd be interesting to see. There are various ways to measure CSF flow in the zebrafish to see if genes that you're interested in are affecting that, and whether or not they develop some sort of scoliotic phenotype. And then you can also stain their bone with alizarin red to look at any sort of bony deficits or abnormalities. And then lastly, our lab is very interested as human geneticists to understand the role of all genetic variation in various genes that we find associated with complex disorders more fully. So often people simply do a one variant at a time, try and figure out whether or not the variant they find in one patient is affecting protein function and therefore is pathogenic and causative in that case. But often what we see is many, many unique variants across a large cohort of people that we don't really know what their function is. And so we've developed in the lab various high-throughput functional assays to try and assess more fully the full spectrum of genetic variation in associated genes on protein function. And so that's sort of a secondary aim of what we're going to do with some of these interesting Chiari genes if we can replicate them and prove that they're truly contributing to Chiari risk. So in conclusion, we find some interesting common variants associated with Chiari, some interesting rare variants across these CHD chromodomain genes in Chiari and Syringomyelia, and that we hope to go forward finding more variants by expanding our cohort, collaborating with as many of you as possible to collect DNA on these relatively rare patients that we then can go forward and try and understand the pathophysiology of this disease a little more through genetics. So future directions, collect more and more. And we need all of the patients to do powerful genetic association tests. We'd like to replicate these associations, so even knowing if these associations that we're seeing are real requires more samples to test these things. And then finally, follow up with interesting zebrafish models and in vitro. So hopefully I've convinced you that we can find interesting things by testing, by sequencing these patients and a large number of controls and looking at the genome. So with that, taking the questions.